Welcome, everybody. My name is Oliver Fien. I am director of the West Coast Metabolomics Center here at Davis, and we organize a monthly speaker series. So welcome to the January 2026 speaker webinar, where we are very glad to have today with us Dr. Julijana Ivanisevic. She is a senior lecturer and head of metabolomics, lipidomics at the University of Lausanne in Switzerland. She did receive her PhD in chemical biology from Aix-Marseille University in France in 2011, before she joined the Scripps Research Institute in La Jolla by Dr. Gary Justak and in La Jolla, Southern California. In 2015 for a postdoctoral training. She joined then University of Lausanne in 2015, to become the head of metabolomics and lipidomics and senior lecturer at the Faculty of Biology and Medicine. Her team focuses on new analytical solutions for both polar and lipophilic metabolite annotation and quantification, and using that and apply these tools to human population and clinical intervention studies. The lab's goal is to advance knowledge of molecular mechanisms that define and maintain cardiometabolic health with reproductive aging. So with these words, I welcome everybody who has joined us, and very thankful for Julijana Ivanisevic, to give a talk today. Julijana, the floor is yours.
So hello, everyone. Thank you very much, Oliver, for your nice introduction. And also thank you really to you and your team for your kind invitation. I'm really pleased to have this opportunity to present today how my team is contributing to revealing the potential of lipidomics for population health research. And so this includes, as you highlighted, the human population epidemiological studies, but also the clinical intervention studies. So I will first start by briefly introducing the problematics and then really focus on the analytical approach we have recently developed for lipid measurement at a very large scale, and this both in terms of sample size, but also the lipid coverage. And finally, towards the end, I will share with you some preliminary results we have obtained so far from the profiling of the Lausanne population here in Switzerland, which clearly demonstrate the sex specificity and the individuality of our circulatory lipid signatures. So why the time is today right to integrate lipidomics as a powerful phenotyping tool in epidemiological studies? Well, ever since the Framingham heart study started, so today more than 75 years ago already, and this strong link between our circulatory cholesterol and the heart disease risk was established, the lipid measurement has really become and remained the mainstay of cardiometabolic risk assessment. And although the bulk components of plasma lipidomes, such as the total triglycerides, free fatty acids, and cholesterol, still remain the keystones of clinical chemistry diagnostics, today using mass spectrometry-based lipidomics approaches, as we all know, we can really measure many more lipid classes, and this in many more details. And this scheme here is really just to remind you about the lipidome coverage we can achieve in human plasma. So this scheme is actually a result of one of the first interlaboratory efforts to characterize this NIST plasma reference material. And as you can appreciate, more than 1,500 unique lipid species were reported and measured at that time during this interlaboratory effort to characterize NIST. So this was done combining multiple technological platforms, multiple methodologies, but also modes of analysis. And over the past almost 2 decades already, these diverse mass spectrometry-based methods have been used in hundreds of cross-sectional, typically case-control studies trying to evaluate how the plasma lipid composition is associated with various health and disease states. So the main aim was, in a way, to discover new biomarkers for diverse cardiometabolic disease states. And indeed, multiple lipid classes far beyond triacylglycerols and cholesterol have been found to be strongly positively or negatively, as you can see from this scheme, associated with our cardiometabolic health, here represented by the cardiorespiratory fitness as really one of the most integrative measures of our overall physiological function, but also a well-established predictor of all-cause mortality. And depending on their biological roles in main metabolic processes, such as the energy storage, the maintenance of membrane fluidity or integrity, but also multiple signaling processes such as in the regulation of inflammation, immune response, insulin signaling, etc. The lipid, the accumulation, or actually more accurately, the changes in lipid levels can indeed have either beneficial or deleterious effect on our cardiometabolic health. And importantly, this effect is not only lipid class, but it's actually lipid species dependent. And today it's very well recognized that even structurally very closely related species, so including different stereoisomers, can have different biological roles and be associated with distinct metabolic consequences. And this is also one of the main reasons why analyzing lipids with the greatest selectivity possible is gaining more and more interest. And one of the greatest examples actually of lipids with deleterious effect on our health are specific species of ceramides, which have been identified as both diabetogenic and atherogenic, and this particularly when they accumulate chronically, so over longer periods of time, due to excessive calorie intake or accompanied inflammation. However, and despite of the fact that the ceramide score has already been introduced into some private clinics, such as, for example, the Mayo Clinic in US, we still lack sufficient understanding about their molecular mechanisms of action. And this is not true only for ceramides, but also for many other lipid species, which have also been significantly associated, so positively or negatively, with the cardiometabolic risk. And so what's really next, and what do we need today to move or to start moving at least the lipidomics towards clinics? So in my opinion, there is really a need to generate more data at different layers, so including clinical gene, protein, lipid data through large-scale, ideally longitudinal population studies and clinical intervention studies. So this is really necessary, firstly, to evaluate the biological variability, and this prior to associating lipids as markers of any disease. Then we need to really identify the link between specific lipid signatures and the actionable phenotypes or different disease stages, which will also allow us to further improve the understanding of molecular mechanisms that are actually behind the disease onset and development. And finally, these clinically interpretable signatures must be validated or actually cross-validated across different populations worldwide, but also different disease subtypes. And finally, the ultimate goal is really to demonstrate the clinical utility by showing indeed the improved diagnosis, prognosis, or even therapy when using these lipid signatures in risk prediction or diagnostics. So this is really one of the main reasons why we became recently, though, involved in several epidemiological studies. So essentially really to collect the critical amount of this lacking data on lipids and steroids in this case, to complement really the existing data that has already been obtained through the clinical phenotyping and the genotyping. And this is really essential because lipid and steroid profiles represent actually the endophenotypes or phenotypes at the molecular level, which are genetically less complex than clinical outcomes, but still equally heritable. And as such, they can actually help us to understand the biology behind the already revealed associations between different gene variants, so genetic polymorphisms, and the clinical outcomes. And this is also nicely supported by the fact that the many genetic loci associated with blood lipid levels are also often associated with the disease traits. And the important point I would like to make here is really the need for the prospective and the longitudinal studies, because with the cross-sectional case control studies, we cannot identify the prognostic markers nor infer the causality between lipid levels and the clinical outcomes. However, the cross-sectional studies, such as, for example, this clinically healthy cohort, are still very useful to study the biological variability and especially to find out what actually discriminates the healthy aging from the disease. But this type of studies can also serve us further to determine also the age and sex-specific reference intervals, and this at least for a subset of lipids, such as, for example, ceramides, for which the internal standards are commercially available and the analytical consensus values have already been established. And as you can see on the example of this specific species of ceramides, the ceramide serum levels definitively depend on age, and this really nicely highlights the need for the age-specific values when assessing the cardiometabolic risk with sphingolipid-based scores. And beyond these population studies for more causal and more mechanistic insights, we are also involved in several clinical intervention studies, among which one of the largest ones is currently the Human Exersome Initiative led by Professor Aaron Bagish. So the main goal of this long-term study is to determine the optimal dose of exercise. So this including the duration, intensity, modality, but also the frequency of exercise that is needed to elicit the health benefits. And these benefits will be assessed using multiple validated health markers, but then also the lipidomic, metabolomic, proteomic, and the transcriptomic data. And in this first initial stage, initial phase of the study, we are really focusing exclusively on the healthy subjects. As you can see, the 20 apparently healthy adults aged between 20 and 40, which are which we hope through the exposure to different types of exercise, different intensities and duration of exercise, will allow us to establish the dose-response relationship between the exercise and the blood concentration of responsive lipids, but also the proteins and some other polar or moderately polar metabolites. Further on, this in later stages, this initiative will also include the older people with confirmed disease in order to really better understand how the age, the ethnicity, but also the presence of disease impact our response really to exercise. Beyond this HEY initiative, we are also involved in a SphingoFit study, which focuses on the effect of the high-intensity interval training in people at elevated cardiometabolic risk. So here we are specifically interested to determine whether the exercise at relatively high and regular dose, so in this case 3 times 45 minutes per week, can really be used to modulate the sphingolipid metabolism. So we are currently acquiring the complete sphingolipid profile, which will then be associated with the validated health markers to kind of better understand really the correlation between the, so for example, the cardiorespiratory fitness, insulin resistance, but also like, for example, here retinal vessel diameters and the sphingolipid concentrations in blood, so before and after the exposure to the exercise. So finally, I'm now getting back to our main study of the Lausanne population with which it all started. So this is the study we are really leading in collaboration with the clinicians, so the clinical consortium here at the University Hospital and the data scientists, and for which we have really invested a significant amount of time to develop a reliable pipeline for lipid and steroid measurement. So as you can see, this study follows the apparently healthy participants, so aged 40 and plus, over a 10-year period. And the main aims of this study are firstly to evaluate the biological variability and hopefully establish the biologically relevant healthy baseline levels, then to identify the associations and hopefully causal relationships between individual lipid species, lipid pathways, and the clinical outcomes. So in this case, we are more focusing on the changes in specific cardiometabolic risk factors that are usually measured in clinics. And the ultimate goal is really to evaluate the predictive capacity of these lipid signatures for improved risk assessment, and this independently or in addition to the traditional risk factors. So as you can see overall, this study involves the analysis of more than 7,000 samples, which have been collected over the two follow-ups or 10 years overall. And to be able really to acquire these valuable complex lipid signatures and steroid profiles with high specificity, but also in a very reliable manner, we first focused on the optimization of the lipid analysis pipeline, and this really with the aim to minimize the technical variance for the maximum of the statistical power later on. And this is one of the reasons why we have opted for a targeted approach while still trying to keep it at an omics scale. So our aim was really to measure several hundred and up to thousands of known lipid species in a straightforward way with high robustness, sensitivity, and as wide dynamic range as possible. And so this decision was really driven also by still multiple unmet challenges of the untargeted approach, and this particularly the limited sensitivity, but also the time-consuming annotation of well-known lipids. And this is not to mention, of course, the inability to appropriately correct for the metrics effects as well. And the development of this high coverage targeted approach has also allowed us in a way to bridge the gap between these two extremes I tried to illustrate. So on one side, the targeted approach, which typically focused before, at least the traditional targeted approach, which typically focused on one pathway or one chemical class of metabolites of interest. And on the other extreme, the untargeted approach, with which we simply tend to detect actually as many metabolites or lipids as possible. And so to enable really the application of the detailed lipid analysis in this large-scale clinical population studies, we developed a single-step sample preparation method, which was coupled with a targeted high-leak MSMS approach for complex lipid quantification. So the aim of this single-step sample preparation was really to maximize the reproducibility, but also the ease of automation, while the main goal really of the targeted quantification using high-leak was to expand the lipid coverage, but also to enhance the quantification accuracy. And we opted really for HiLiP due to its ability to separate lipids by class, so based on their head groups. So in these graphics, you can see really the lipid classes that we can detect in positive and then also in negative ionization mode. To enhance really the quantification accuracy, we use the Ultimate Splash 1 mixture, which contains overall 69 internal standards, and we have added 5 more additional standards in order to cover some lipid classes for which we were the standards were not available in the mixture itself. So this means that we actually have multiple. I'm sorry. It's okay. So this means that we actually have among these 75 internal standards, multiple representatives per each lipid class, and this with varied fatty acid composition, so varied chain length and the degree of unsaturation. And importantly here, this lipid class separation using HiLiP, so based on head group polarity, was really an important advantage because we really wanted the endogenous lipids to collude with the corresponding internal standards, and in this way allow us for the appropriate correction of matrix effects under the same solvent composition. And to maximize the method coverage, we have implemented the strategy with an initial qualitative screen. So for this screen, we have assembled as many transitions as possible. So based on the detectable lipid species in human plasma, serum, whole blood, different types of blood cells, and different tissue extracts. So as you can see today, the method includes the predefined SRAM transitions for over 2,400 lipid species. So these transitions are distributed across 4 different methods in positive and 4 different methods in negative ionization mode. And this was really done to ensure the optimal peak definition. And following this initial screen, which is usually performed on the pooled samples, which are representative of each sample group, the signals are then filtered based on the several criteria that you can see in the middle. And this is really to ensure that only really robustly detected species across all replicates of pooled samples can be later on quantified. As you can see on this example of the NIST plasma reference material, we are quantifying here so more than 800 lipid species in plasma in a total analysis time of 12 minutes. And this combining, of course, the positive and the negative ionization mode. So this was made possible by a dual column setup, so supported by these 2 pumping systems. As you can see it illustrated here on the Vanquish Horizon Duo from Thermo. So here I would really like you to note that despite of the fact that a very high number of transitions are of course targeted in specific areas of the gradient, such as, for example, the void volume, where many neutral lipids collude, we were still able to achieve a very good peak definition with a minimum of 8 to 10 scans at least per peak, at least in this area, of course, where the coelution, so specifically of neutral lipids, is very high. In the other areas of the chromatographic gradient, we have a much better peak definition. And so this was made possible due to really the superior scanning speed of the TSQ Altis instrument. And we could clearly bring down the dwell time to only 0.25 milliseconds, as you can see right. And Overall, with this developed pipeline in human plasma, we were able to detect more than 900 different lipid species in total. So overall, we cover 9 different classes in positive ionization mode. So this includes essentially the neutral lipids, but also different classes or subclasses of sphingolipids. And we have also recently added the acylcarnitines. In negative ionization mode, we cover around 11 lipid classes. This includes all different subclasses of ester and ether-linked glycerophospholipids, but also the lysophospholipid. And we have also recently added the tissue-specific cardiolipids. We are currently further upgrading this method to achieve the more selective quantification with a fully resolved fatty acid composition, at least for some lipid classes or specific lipid classes for which we have not achieved it yet. So this was made possible with the release of a new hybrid quadruple ion trap mass spectrometer, which is called Stellar, so released, I think, last year at the ACMS, with which we can acquire the full MSMS spectra in different fragmentation modes. And here you can see really this this allows, for example, on this sphingomyelin example, the more selective sphingomyelin characterization by combining actually the fragmentation patterns in HCD and CAD modes. So as you can see in CAD mode, we can actually acquire the product ions that are specific to the sphingoid backbone that then allows us to really determine whether it's indeed the sphingosine or maybe sphingonin, sphingadienine, or some other long chain base. And the same is true also for 3-acylglycerols, which can be thanks to, using Stellar, more selectively quantified at the MS2-3 level to really provide the complete fatty acid characterization, or to achieve really the complete fatty acid characterization. So for now, we are currently only testing the applicability really of this approach, simply first for the qualitative characterization of the TGs in the human breast milk. The quantification will come afterwards because, as you can imagine, the association with internal standards is a very complex task. Finally, back to the current method so that we have really running on the TSQ Altis. As you can see, the measured concentrations of lipids in the NIST plasma reference material span more than 6 orders of magnitude, so really ranging from this very low abundant PGs and hexosyl ceramides to a really more, highly abundant cholesterol esters and other neutral lipids. We have also carried out the accuracy cross comparison with direct infusion approach using lipidizer. And as you can see, the high correlation between we have observed a high correlation between measured concentrations, with the exception of some 3-acylglycerols and fatty acids. So overall, the greater diversity of lipid species was covered with a high-lick approach, which is in a way justified by this added value of the chromatographic separation. And this was specifically an advantage for the detection and measurement of the more some low abundant sphingolipids and phospholipids. And here you can also appreciate the match between the quantified values of ceramides in NIST plasma. So using either our high-lick workflow here in orange and the consensus values that have been actually established as a result of the ring trial, so here in blue. So this is just simply to provide you an idea, as a kind of even stronger argument for the accuracy of our method. We were really happy to see this, that even with this global lipidomics method, we can achieve a very comparable results that very nicely match the consensus values that have been obtained through the ring trial. So following the accuracy cross comparison, we have also benchmarked our method by the characterization of the entire suite of NIST materials, so the materials with different clinical and ethnic backgrounds. So as you can see, the acquired lipid signatures really allowed for a straightforward distinction between these different plasma materials. And as you can observe in the high TG plasma, so which was collected from the patients with hypertriglyceridemia, for example, the significantly higher levels of triglycerides were observed in more than 1/3 of the measured TG species. And here I would really like to make an important point that these differences in lipid composition between plasma materials that are collected from individuals with different clinical backgrounds can lead to significant differences in matrix effects, of course. And I try to show this really in the bar plots on the right, where you can see that the signal of the triglyceride internal standard was suppressed by more than 40% in the high TG material compared to the other plasma samples. So here it's really important to consider that these differences should be corrected with the internal standard spike if we indeed wish to reveal the subtle but physiologically relevant changes within the heterogeneous human populations. So this is also one of the reasons why we are primarily oriented towards the more quantitative targeted approaches when it comes to really clinical or human population studies. And beyond the use of the internal standard spike, the authenticity of the internal standards is equally important, and this particularly when the endogenous lipids do not collude with the internal standards. So here you can see the results of the ceramide measurements using both authentic, so in gray, or the surrogate internal standards in blue for these two different ceramide species. So this was done in a context of the interlaboratory and cross-platform ceramide trial, which was led by the Singapore Lipidomics Incubator. And the measurements were performed either according to the SOP, which was distributed among us, or using other existing methods that were available in each respective lab. And as you can see nicely, the concordance of the results between labs was actually not affected by the method itself. So the values generated by SOP group, so here in red, versus other methods, so here in blue, were not actually significantly different. And it's only really the calibration using authentic labeled standards that allowed for the significant reduction of the data variability. And so this here from up to 60 down to only 10% for this ceramide species. So this nicely really confirms that the authentic internal standards are actually the only or the best strategy, of course, to correct really for the differential also ionization and fragmentation efficiency. And this is really something very important to consider, particularly for when it comes to the lipid measurement, and especially the measurement of neutral lipids more than other lipid classes. So following really this parenthesis on the importance of the internal standards, I will now share with you some preliminary results we have obtained so far from the profiling of the Lausanne population. So as I previously mentioned, at the baseline, we selected around 2,600 apparently healthy participants, which were followed over 10 years' time. And in the described methodological setting, with the automated sample preparation, we can analyze around 5 batches per week. So this means more than 400 samples overall, and this including the internal and external QCs for the continuous quality assessment. And as you can see here, we were able to measure with quite high precision, so the CV below 20% in each individual sample, a wide panel of more than 780 lipid species over the course of analysis of these first 13 batches, so more than 1,000 samples overall. So as you can observe, the average analytical variability was below 20% across all lipid classes, with the exception here of phosphatidylserines, which could barely be detected in NIST plasma. So their very low signals clearly had an important impact on the signal, on the variability, I mean the measurement variability. And on the top of the analytical variability, which is here again illustrated in white box plots, we also explored further the biological, so the intra and the inter-individual variability. And as you can see, the intra-individual variation was consistently lower than the intra-individual variation, as expected, I would say. But this also nicely suggests that the individual lipid profiles are actually highly distinct or personalized and stable over time. And interestingly, it's really the TGs that have shown the highest degree of intra and inter-individual variability. So here you can observe this really at the species level. So the bar plots here show really the intra-individual, and the lollipops, this inter-individual variability. So this highest degree of variability in TGs, for TGs, is likely a reflection of our lifestyle, I mean differences in our lifestyle exposures, and highly likely, I mean probably specifically the dietary, the differences in dietary regimens. Overall, this very high level of individuality of our circulatory lipid signatures was also confirmed using the unsupervised multivariate analysis. And here you can see the results of the hierarchical clustering analysis, which show the distances in lipidome for plasma samples from 20 randomly selected participants across all 3 follow-ups, so overall 60 samples. So please consider that the similar results were obtained through successive resampling across different individuals, of course. And as you can see, so here highlighted in different color tones, the lipid signatures indeed show a very high level of individuality, with the samples from the same individual clustering together, and this at the time scale of 10 years. So this was quite surprising to me, I mean, to see that even over 10 years, these lipid signatures will have the tendency really to cluster together. So when they are have been acquired from the same individuals. And these results are also coherent with the recent results of yet another independent longitudinal study, which was done by Professor Snyder group in US. So they could also observe that in comparison to our transcriptome proteome, but also the polar metabolome, the lipid signatures appear to be much more highly personalized, so reflecting really our chemical individuality, so both the genetic polymorphisms, of course, but also our unique lifestyle exposures. So this is also one of the reasons why today they constitute such a valuable source of biomarkers, so likely really capturing the impact of various lifestyle factors. And within these acquired lipid signatures, beyond just this high individuality, we could also observe numerous significant sex differences. So as you can see, more than 50% of measured lipids displayed a very significant sex differences with, for example, the sphingomyelins and phosphatidylcholine, specifically with polyunsaturated fatty acids in their composition, were found to be present in significantly higher concentrations in female plasma, while in contrast, males showed really the higher concentrations of TGs and some specific species of lysophospholipid. And while the origin actually of these differences remains, of course, to be more investigated, it is hypothesized that they are driven by sex hormones. They are the master regulators of our lipid metabolism, after all. But they also, they also, in analogy, are a result of a differential activity of specific enzymes, such as, for example, the phospholipases and fatty acid desaturases that are involved in sphingolipid, etherophospholipid, and glycerolipid metabolism. And just to briefly conclude, all these results taken together, I think, nicely highlight really the need for the determination of stratified and, of course, if possible, in future, the personalized reference intervals. And they also really strongly suggest or fostered further investigation and consideration of sex differences for cardiometabolic risk assessment, for clinical diagnostics, and of course, for treatment. And with this, I would like to, my special thanks actually go to my really motivated, skilled, and dedicated team here in Lausanne. So as you can see, we are trying to look after our cardiorespiratory fitness, but not only our nutrition as well. And many thanks, of course, to our collaborators. So Thermo Fisher team, triple quadruple team in San Jose, the clinical consortium of Colaus and Psycholaus, clinical consortium of Complete Health as well, the SLINK team for the help with the isotopic overlap correction, the Martin Gira's team at Leiden University for the cross validation of the method accuracy, Gonzalo at the Imperial College for the help with statistical analysis, and I hope I haven't forgotten anyone. And yes, I will just wrap up this talk with this quote from my team. So please keep enjoying Untargeted, but yes, don't forget to appreciate the power of targeted analysis. I must say that as we have a very heavy load of services, I don't know how it's on your side, but we have observed a significant shift towards more application of more targeted quantification analysis, as compared to the trendy, previously very trendy application of untargeted approaches. So yes, thank you very much for your kind attention and for invitation, of course.
Thank you so much. It was a very good presentation, very interesting to see what you have achieved, and the first glimpses on large-scale epidemiological studies. I would like to open this presentation to discussion and questions. So, Shen, would you like to just open your mic and have the first couple questions?
Yes, I do. Perfect. For the quantification that you have done with the HELIC, is it only based on the single point normalization, or you actually run curve or any other more advanced approaches you tried?
It is based fully on the single point calibration, correct? Yes.
And during the ring trial, did any other lab, their in-house method is using more advanced and more like single point and show any advances in the result?
So you might, I think the best example, the best comparison was done in this ceramide ring trial that so the results were published actually last year in Nature Communications, and there they really compared let's say the accuracy of results using single point versus multiple point calibration. So it's really multiple point calibration curves, external calibration curves, in addition to the internal standard spike. And surprisingly and interestingly, they have not observed any significant impact. Let's say improvement by using the external multiple point calibration curves. Yeah, for ceramide. Specifically quantification. Ceramides are a good example because we do have authentic internal standards. Yeah. Which is not the case for any other lipids. Yeah.
Yeah. Now I'm pivoting to the stellar method. Is the HCD and CAD fragmentation happen at the same time, simultaneously in the same method?
No, this is done subsequently, yes, not simultaneously.
Yeah. Does the method or the transition time doesn't allow it, or what is the limitation?
So to be very honest, we have not had a chance to test it yet in-house because we have simply been performing some tests remotely. So the thermo team has been performing some tests in San Jose. So we are going to get stellar by the end of January. And then I'm very happy to answer your questions in more details. Now it would not be fair, to... Because we haven't had ourselves the hands on the instrument yet. Yeah.
Because there's basically the lower mass is not captured by the CAD, so the less abundant fragments show up.
Yes.
Higher.
CAD is really a softer fragmentation, if you want. You can get, interestingly, not only for sphingomyelins, some advantages have been also seen for the bile acids, for example. This is also very interesting because we can really fragment the parts of the molecules that otherwise, we could clearly not get the specific fragments with the HCD.
Yeah, it's going to be super low. So at the end, do they have a solution to stitch the two information together? Because we don't want either or, we probably want both.
Yeah, this is a very good question. I'm waiting to see how are we gonna perform the data processing and deconvolute all this data. Yeah, you are right. This is not going to be trivial.
Thank you. So Josiane Pavot has the following question. In these studies, could you correlate the cardiolipin quantification with cardiovascular risk?
So this is a very good question. We have not, so in plasma, we have not detected the cardiolipins. They are a priori the tissue-specific lipids. You said cardiolipins, no?
Yes, that's what Josiane asked.
Yeah.
So no cardiolipins in plasma, okay?
No. But we have detected them, of course, in different tissue extracts. Yeah. But I cannot say more on the associations. Association with the cardiometabolic risk.
So then I have another question myself here. If you say that the biggest lipid variance is in between people. And it might be due to diet. So people have different types of food and, therefore they have different types of triglycerides. And that's fine. That's very easy to understand. But there must be also a genetic component. Lots of GWAS studies have shown genetic components. Do we then really need the lipidomics if the genetics itself could also already explain things?
Well, for sure, lipid levels are genetically determined, but they are not only genetically determined. That's for sure not. So, they are genetically determined only to a certain extent. I think the impact of our lifestyle exposures is also significant, if not as significant as the contribution that is coming from our gene variants.
So then I have another question. If if there's so much interpersonal variance, and
Yeah.
is contributed by both genetics and by diet and or other types of exposures like exercise and you name it, could it be that it's the genetic component at least is not just the biosynthesis but also the packaging in lipoproteins? So what I'm missing here is a lipoprotein fractionation based cohort study where, because, it's blood and there are keratin lipoproteins, and those lipoproteins carry the message. They would bind in extra, hepatic tissues. They would then offload their cargo there. So that is maybe really a missing link in lipidomics. Is this being discussed in the various discussion groups that you are involved in?
Yes.
Lipidomic Society
Yes.
and whatever. Are people looking at that?
This has already been highlighted multiple times, and I mean, you're absolutely right. We would definitively need to perform the lipoprotein profiling using NMR and really, yes, to get the lipoprotein profiles that then can be associated, hopefully, with the results, lipidomics results. Yes, definitively. Yeah, no, this is a missing link. I'm not denying this at all. An important missing link. Yeah. Yeah, as you said, I mean, this is how lipids are carried in blood. They are clearly not soluble in plasma, in aqueous plasma. So yeah, they are incorporated within the lipoprotein particles. Yeah.
So do we have other questions by other people on either technology? Yeah, Shen?
The downstream analysis, rather than correlation, is there something else that's brewing, like to dive into the causal effect?
Yeah. So so ideally, so this is as I try to, I mean, as I mentioned, the study is really done in collaboration with the statistical geneticists who are involved in multiple genome-wide association studies. And the idea is, of course, to use the data from other bigger population studies, such as the FINRISC or the UK population, from which we can get out the previously established associations between the gene variants and the lipid levels. So we clearly don't have enough, don't have enough big sample size here to perform any, to try to reveal any new associations between the gene variants and the lipid blood levels. But the causality we can also look at the causality, because this is the longitudinal study, and we have acquired the lipids and the steroid data not only at the baseline but also at all different follow-ups, the same as all other data layers. So you see, we will be able to look into how the changes in lipid levels are correlated or potentially associated with the changes in the cardiometabolic risk, specific cardiometabolic risk factors that are measured in clinics.
Like, do we see, like, typically clinically, we're gonna test VLDL cholesterol, like those, or HDL cholesterol? I think that might be part of the panel. Like, do you see
Yes, but also all the markers of diabetes, such as the glucose tolerance, the HOMA index for the insulin sensitivity, then the adiposity markers, yes. So yeah, we will, there's some inflammation markers as well that have been collected through the clinical, routine blood analysis. Also, the physiological parameters such as the blood pressure. So yeah, all these different clinical kind of standard clinical parameters will be considered.
One last question. You mentioned the steroids panel. Like, can you elaborate on that? How do you measure it reliably in plasma?
Yes.
With a special caveat to that?
Yeah, for the steroid analysis, we have developed a completely separate method, so reverse phase method, which is fully covered by internal standards, meaning that every single steroid has its own associated, authentic internal standard, which, as you imagine, is the only way to kind of get down to really absolute concentrations. So yeah, this is done really separately, and it's much more fastidious, much less high throughput, let's say it this way, than this global lipid analysis. So this is still ongoing. We have started recently, but yeah, we have a 30-minute method in 20-minute method in positive and then 20-minute method in negative ionization to cover a panel of 22 steroids overall. So this includes the sex hormones and the corticosteroids.
Yeah, do you have to derivatize them, or you require big volume to reach a good, decent sensitivity?
No, we do not derivatize them. We use the SP to pre-concentrate the sample, and this works quite nicely. And then we use the ammonium fluoride in the mobile phases in negative ionization mode to enhance the sensitivity, specifically for some estrogens and aldosterone. Yeah. But actually, we start only from 100 microliters of plasma, so not more than that.
All right.
Then, of course, yes, this is limitation. This is a limiting factor, but we didn't have more material than this because it's quite precious. Yeah, the aliquots are quite precious.
There's an alternative method by Biocrates, who offer a kit for steroids. They have been commercialized in Europe, and they were they couldn't be commercialized in the United States. So have you compared your steroid kit or your steroid method against the Biocrates kit? And what are your thoughts about that?
So, no, we haven't, but you gave me a good idea because why not? We can do it. We are just in the process of kind of putting this methodological manuscript together where we evaluated the accuracy and precision of this steroid method using Chrome systems as a quality controls. And we can. It's still time to do it. So why not? I'm now curious. I was not aware they released this specific kit for steroids.
Oh, long years. So for many years.
Okay.
Many years.
Okay.
Only in the United States there were patent issues. That's why they couldn't do it in the United States, but in Europe it was always possible.
Okay, and this covers quite a wide panel of steroids, or
we have the panel. We have it actually in-house. So now the patent has expired. So we are testing that right now in our own lab here, actually on the Altus instrument. So that's yay.
Okay.
And some people in my lab, at our labs, are doing it right now. So I just wanted to see if you already have a comparison, because I'm always a little skeptical on kits. And also 17, Uri Keshet, our lab manager or director, said so. There is a brochure. He also put it into the chat box. So the 17 steroids, but they also use C18 methods for cleanup and whatnot. So there is some commonalities, what you just said. And I'm always a little skeptical, or I'd like to see cross validations between methods, right? Just as you have shown for ring trials, and so on. I also have seen, for example, that other people do large scale MRMs. Like the Metabolomics Innovation Center in Canada offers a kit was 2,000 pounds. And
I know.
And many of them are just infusion based. So what do you think about those methods?
I fully agree with you. Huh? I
haven't said anything. I have not made a I just.
No. You said you are skeptical.
Okay, skeptical. Okay.
You said you are skeptical about kits. Well, at least Biocrates kits. we were in a way purchased by Biocrates to implement their kids for our services for several years. I cannot, so we never got to really test them, if you want, because I kind of always refused to use them, mainly due to the cost problems, if you want, because with our in-house methods, we can significantly reduce the cost compared to the cost of the kids. So that was my primary reason, if you want, at that time. Then I was very skeptical about this acyl carnitine. I think this is, yes, the acyl carnitine quantification that is done with direct infusion analysis. Yeah. And I think there are some people proved the problems. Actually, this was just published in January this year by Professor Wishart. I think they cross-compared their method for acyl carnitine quantification with the Biocrates kit. If I'm not wrong, look into this.
Acyl carnitines are always used in inborn errors of diseases for decades. So acyl carnitines have permanent cations, and they are easily ionized, so they have much less ion suppression. So I'm not skeptical on these compounds, actually, not so much at least. But I also see that people like Dr. Wishart, they say we have 2,000 compounds, many of them are lipids, and we use direct infusion. And I always thought that there's too much overlap. There's maybe even in your cases, you had Hillick methods and as well as reversed phase for triglycerides. So if they're not chromatographically separated, you have so much overlap between abundant lipids, and then there are isotopes with less abundant lipids that have maybe a double bond somewhere or not, right? So I don't know.
Yeah. No, it has to be. I mean, the data has to be corrected for the isotopic overlap. Absolutely. So this is done with the Sleeker application that was developed by Slink, so by Amaury Kaznav group. Yeah, you're right. I mean, but of course, we cannot have with Hillick, we cannot possibly resolve the isomers. I mean, this is not even. This is not the aim. In this case, yes, this is more used as a global lipidomics method for more for a discovery, you see. I mean, discovery in terms of associations. Then, of course, if we find some very interesting lipid signatures, we will focus on them with a very specific and optimized targeted methods. That's without, I mean, we cannot possibly think about this method going to clinics. That's, I mean, to me at least, that's something to consider. It's, yes, clearly used for the research, despite of the fact it's a clinical research.
So you showed also a paper by Peter Meikle from Australia. And so Peter Meikle has, a targeted method, multi-targeted method based on reversed phase for hundreds of lipids. Why didn't you just adopt that one?
I can tell you one reason. We found it extremely fastidious to integrate all these peaks. It's really tough. I mean, on the reverse phase, you would be surprised because they can distinguish even between the specific ether and ester linked species for the phospholipids. It's I believe a very fastidious data processing. And then, so why? Also because simply we are more high throughput with this method. I mean, it's 12 minutes combining positive and negative ionization mode per sample. I think they are at least 20 minutes or more. If I'm not mistaken, I don't want to claim something because I don't remember exactly. yeah, these are the main reasons. I mean, and then no, of course, the one of the most prominent reasons is the coelution of endogenous lipids with internal standards. We really thought this is one of the greatest advantages of HILIC. Yeah, to enhance the accuracy of quantification. I mean, the ability to correct for the matrix effects. That's a huge issue. That's a really huge issue on reverse phase.
Okay.
I believe so.
So are there further questions by the participants? I see many people are pretty quiet here. So we have dominated the discussion a little bit. We apologize for that, but great discussion, good answers. Are there further questions by other people? If that is not the case, then I would like to thank Julijana. That was a fantastic insight into what's going on and over there, Switzerland. I hope that we will visit you and also go skiing and having fun at some point.
That would be great. Yes,
yeah.
We should organize something. We will think about something.
Yeah, make a big meeting. I have to go to Switzerland. All right. Thank you so much for joining us here. Thank everybody else who joined us. So next month we will have a speaker from Cornell University, Jan Krumsiek, who will talk about multi-omics integration. So in case you are interested, please join us back for next year's hybrid seminar. So it's both in person and by web. Thank you so much, everybody.
Thank you very much. Bye-bye.